are seeing me after almost 3 weeks. So, by now we already have almost all the basic components and other parts of immunology, and in last lecture you have heard the hypersensitivity or allergy, and this allergy or what we are going to discuss today is the autoimmunity, that both are actually non-infectious, right. So, it is not from pathogen. So, from the name autoimmunity you can understand that the immune reaction against our own or self, right. So, during the development of T cell, development of B cell or diversity clonal selection hypothesis, we always continuously said that all the auto reacting receptor containing lymphocytes are eliminated, that is the clonal deletion. Okay. Today I am saying that no, it is not completely deleted, not only from our self antigen, but you know that we have lot of microbes or microbiota in different parts of our body, particularly in alimentary canal. So, that microbes are foreign to us, but immune system they do not react with them, right. So, they are so integrated part of our system, the immune system thinks like they are, I mean all the commensal microorganism I am talking about, they are kind of our own part of the body, right. So, autoimmunity when it is developed, like immune reaction against our own or self protein, it is autoimmunity. But if there is any autoimmune reaction or the immune reaction rather against this microbes present in our body different parts, this is called xenoimmunity. But the reaction or the reaction type or the effect or the disease symptoms all are so similar. So, both we are going to talk almost together. Okay. So, you will not differentiate xeno and autoimmunity. So, the basic concept or the basic principle why autoimmunity develop is the major part or the major role of immune system when it is not working properly. Major role of immune system is to identify self and non-self. So, what is the procedure that we so far discussed is that somehow immune system learn to tolerate our own protein, own system, own organ, but if somehow that breaks, okay, so then immune system start reacting against our own protein. In fact, if you consider like when I was telling or other Professor Ganguly was discussing the B cell immunity, and in my lecture I discussed T cell immunity. What happened? We always say that it is not reacting with our own protein, because the self protein recognizing receptors of B and T cells are eliminated, but it is not practically possible. Why? I mean that kind of question might have came to you in your mind, that see there are many proteins in our system which is very similar to the foreign pathogen, it is possible. Second is, I mean very similar, when a foreign pathogen attacks and you know that antigen processing happens. So, the overall protein may not be similar to our own protein, but after processing the piece of peptide that developed, that may be very similar or very very rarely it is identical, but it is possible, that possibility is there. So, if all self reacting antibody or the B cell receptor or the T cell receptor, if it is eliminated from the system, then what will happen? Our immune system will be impaired. Okay. It cannot function properly, because if there is a commonality between a pathogen antigen or our own antigen is similar, if the similar one completely eliminated from the system, then what will happen? That pathogen will also be neglected by the immune system, because they cannot recognize it. So, it is not happen completely, but there is a fantastic balance, so that very rarely we have autoimmune disease. So, that means immune system somehow manage that. If any how or by any means, if that balance is gone or disturbed, then autoimmune disease is happening. Okay. So, first what I will do is we are going to discuss this autoimmune disease in the first part. There are I divided actually in 3 parts. Before that, I am supposed to tell, like, this is the acknowledgement, and most of the slides are taken from this Genoese Immunobiology book, 9th edition, and which is a copyrighted product. And in autoimmunity, we are going to break into 3 parts actually: the making and breaking of self tolerance, that what we are just discussing, like how this self toleration, I mean, we already discussed how this self tolerance grown, or how B cell, T cell develop. So, we are going to discuss again, and then autoimmune disease and pathogenic mechanism, like how autoimmune disease is developed and what are the pathogenic mechanism, how the disease evolved, what is their symptom. Definitely, we are not going to talk like detail about the disease and all the symptoms and things, because this course is not for the medical student. And third, is there any genetic or environmental basis of autoimmunity, so that we can figure it out, okay, this disease is genetic or environmental, or is just accidentally it happened, okay, in any individual. So, first we are going to talk about making and breaking up self tolerance. So, I already told you that critical function of the immune system is to discriminate self and non-self, right. So, identify the self, do not do harm, find the non-self, clear it or clean it from the system. So, this is a big table, okay. You do not have to worry about that. This is what happened. There are different disease in this panel, like psoriasis, rheumatoid arthritis, Graves disease, and Sjogren's syndrome, Crohn's disease, multiple sclerosis, type 1 diabetes. Here is what I am telling you, do not have to remember all the table, like, okay, you can, again I am telling you can, and you can go through it, you will find many information from here. What it is telling is these are the disease, and this is the disease mechanism. So, if you see that it is mostly whatever is written here is either antibody or T cell mediated, right, and this is the consequence what happened to this disease. Okay, say Graves disease, what is happening? It is hyperthyroidism, that means overproduction of the thyroid hormones, okay. And this last column is the prevalence, and it is actually not alphabetically, it is according to the prevalence of the disease. So, you can see that psoriasis is 1 in 50 maximum frequency, and type 1 diabetes is 1 in 100. Okay. That means, this disease we do not see many, but it is not that rare also. Many cases it is very common to many of us. That means, our immune system is not working properly or active in all possible ways correctly in all the individuals. Okay. So, what are, I mean, this is a summary slide what we already discussed in previous all the lectures, okay, except if you just ignore the hypersensitivity, all these are already discussed. How immune system tolerate our own protein or own antigen, because there is a central tolerance. What is that central tolerance? That is the deletion editing. That means, B cell and T cell receptors are B cell and T cell containing receptor which interact with our own protein will be deleted. Okay, that we are telling from the very beginning, clonal deletion method, and this happened where? It happened in thymus and bone marrow. Okay. Then antigen segregation, because all antigen are not exposed to the immune system, like there is a physical barrier, okay. And most of the time what happened, the immune reaction is happened in lymph node or spleen, in the secondary lymphoid organ, and secondary lymphoid organ do not have the access to all the organs, like they do not see the pancreas, they do not see our retina. Okay. So, there are many specified organ where immune system where the reaction happened, actually they do not see each other. Okay. So, like here the thyroid, pancreas, there are many other such organs. Then peripheral energy, that we have already discussed, like even after the central tolerance, when it is eliminated during the development either for B cell in bone marrow or T cell in thymus, after that what happens? They come to the peripheral blood. In peripheral blood or peripheral system also they interact with many cell protein, but if the interaction is not very strong, okay, that we discussed during T cell development. If the interaction is not very strong, if it is weak, then they survive. Okay. If the interaction is very strong, then they die. They die by apoptosis. So, that is clear. So, not only the central tolerance, there is also a peripheral tolerance also. So, after after coming to the periphery, if B cell and T cell can interact with our own protein or own cell, so they are not going to be activated. And if it is a single interaction, just like B cell receptor interacting with the protein only, no T cell is involved, or only T cell receptor interacting like cytotoxic T cell receptor interacting. So, they are supposed to kill it, no, it needs another activation, right. So, the antigen presenting cell should give the co-stimulatory signal to activate. If you remember the T cell activation, there are 3 signals are required, 1, 2, 3, right. There is one signal by T cell MHC, second signal by B7 and CD28, and the third signal by the cytokines. I am repeating it because we discussed long back, you might have forgot that, or even if you remember, I just reminding you again. So, if only one signal 1 is there and 2 and 3 is not there, that means no co-stimulatory signal, that T cell will not be activated. Okay. So, in that case what happen? It become anergic. Anergic means they will be non-responsive forever. So, this is how it is happen, and so peripheral energy is also happening in the secondary lymphoid organ, because interaction between B cell and T cell and antigens are going to happen in the secondary lymphoid organ. There are regulatory T cells that already you know, different subsets of T cells like T17, Treg, and Treg is controlling the activity of other T cells, because it is, that is why it is regulatory. So, most of the time it is a negative regulator, it is repressing or suppressing the T cell activity, and that how it is of all, you know, that signaling is through cytokines. So, they gave the intracellular signals, and the secondary lymphoid tissue and site of inflammation, wherever the other T cell act, the T regulatory cells stop them to be activated. So, that this is whatever I am telling here now is a normal case when immune system working perfectly. Okay. Functional deviation. So, differentiation of regulatory T cells that limit inflammatory cytokine secretion, okay, and secondary lymphoid tissue are the site of inflammation. So, that again they stop the inflammation also, not only that interaction and killing, inflammation is also stopped by the regulatory cell, okay, and activation of induced cell death. So, that we are going to discuss again. So, these are the points I am telling. So, that means if any cell interact, any cell means any B lymphocyte or T lymphocyte interact in the lymph node with our self antigen strongly, that also gets the signal to die or death, okay, by apoptosis. So, that means, and how suddenly it will happen, because initially they are supposed to be deleted within bone marrow or thymus while when they are developing or maturation is happening. But two possibility, one is they can miss that, somehow the interaction was not that strong. So, they missed that screening, came out to the peripheral system, but there is another chance in the lymph node also they have a possibility to interact. Even after that, if it is very strong, then they will die. So, apoptosis induced cell death of what kind of cell death? The B cell or T cell, and it happened by apoptosis, and it is also in the secondary lymphoid organ. So, this is the normal mechanisms by which our immune system works properly. Clear? So, any of this mechanism, whatever I just discussed, any of this mechanism, if it is not working properly, that means autoimmune disease. So, anything anywhere is missed or not working properly is autoimmune disease. So, autoimmune disease whatever the reason I just discussed, how the self tolerance can break any of the previous points, that can happen for two reasons. One, the genetic factors, it may be genetically regulated, or there are some genetic defects are there, or maybe some infection or environmental exposure induce that autoimmunity. Okay. Either genetic factor or infection and environmental exposure, both are, or both or independently alone, individually also can induce the immune dysregulation or dysfunction, which can lead to autoimmunity. Okay. Autoimmunity means development of the disease. So, now we are going to again one by one to see little more detail. Central deletion or inactivation of newly formed lymphocyte is the first checkpoint of the self tolerance, that we already told, right, that you know already so many times, so many ways we discuss that. How this thing happen? Because you see, when this thing happen in bone marrow and thymus, say here are only two examples are given, say retina or ovary. Retina and ovary expressed some specific proteins, many other organ produce specific proteins, say pancreas produce insulin. Okay. It is a pancreas, and not only whole pancreas, there are islets of Langerhans, and beta cell only produce insulin. It is so specific. Now, how a T cell, this is the example of T cell, you can extrapolate to B cell also. How T cell will see insulin and retina specific or the ovary specific proteins in thymus? There are many proteins which develops after puberty, right. Many hormones develop after puberty, many proteins may synthesize after puberty when T cell is almost done. Okay. So, what happens? So, there was a theory, like all the thymus epithelial cell, okay, medullary cells, where the negative selection happened, they express all the proteins. So, they express all the genes transiently, so that during T cell development they can see what is going on, and they expose to all the proteins. And eventually what happened, there was a transcription factors AIRE, which is autoimmune regulator. Gene was discovered later on. Okay. It was found that AIRE actually the transcription factor which regulates the expression of all the proteins in the thymic medullary cells. Okay. In the thymus medulla, where the negative selection happened, it expressed all our own protein. So, what happened? So, during that period, that means time of the negative selection, they exposed to all our own protein. So, whichever is interacting strongly, they die. But normally, if they interact mild, interaction is mild or not that strong or moderate, they ignore it. Otherwise, I just told in the beginning that if they eliminate everything, then our immune system will also be very weak. So, strong binding will die, and only those which is not interacting strongly, they survive and come into peripheral. Then the peripheral tolerance will come. How this discovered? Because the mouse, okay, what they did, they delete this AIRE. Okay. If the AIRE they deleted, it develops autoimmune disease. In human, you cannot do that, okay, but there are disease. Okay. If by any chance genetically, if this gene is not acting properly or expressing properly, what can happen? It can happen, you can see autoimmune polyendocrinopathy, candidiasis, ectodermal dystrophy. So, this is a very big name, APECED. No, you do not have to remember all this, you can, but you think that this is how each gene or each function. So, when any gene function was discovered, and we have to find for human, we have to find is there any individual where this gene is not there and see what is happening. Okay. So, this is autoimmune disease, and now after this when AIRE, but we have a very handy model, we already discussed the mouse, we can make a knockout mouse. So, we make a mouse, but this gene is not there and see what is happening. Okay. But incidentally what happened, we have some human which is defective AIRE, and this autoimmune polyendocrinopathy, candidiasis, ectodermal dystrophy kind of disease, their symptoms and many these things we do not have to remember, okay, or I did not put in the slides also, because again I am telling we are not You are mostly not the medical students, and I am also not a doctor to explain all these things, and this is not also scope of this study. Then second point, lymphocyte that bind self antigen with relatively low affinity usually ignore them, but in some circumstances become activated. So, first normally they ignore, so it is normal, but if they activated, that means it is not wanted. I mean, after coming to the peripheral blood, if they interact, very mild interaction is happening, because there are some similarities always possible, because anyway they are all are proteins. So, maybe out of say 8 amino acids, 6 are matching, 2 are not matching. So, interaction, if 8 are matching with the epitope, the interaction will be very strong or nice or good. But if it is 6 or 5 are matching, it is not that 0 interaction, there will be an interaction, because protein protein interaction does not, they do not know what is the fate. So, that moderate or no low affinity interaction normally ignore, but sometimes it get activated. How? See here, this is an example. So, wherever example is possible, I am going to give you the example. Here what is that? B cell specific with specificity of DNA. So, if B cell receptor, okay, you also know that this receptor dimerization is very much needed. It is not single receptor interaction is not always activate the cell. So, they cross link the receptor by DNA molecule is big. So, if DNA molecule internalize, what happen? There are TLR9 which has this CpG or GC rich actually methylated version of DNA gets activated. What happened? The immune system is not made for that. Immune system is actually the methylated eukaryotic systems is eukaryotic DNAs are methylated mostly, but prokaryotic DNA are that not, I mean in the GC rich region. Okay. So, immune system made for any DNA, if it is non methylated, that they will bind and this TLR9 will act. But in some case, if the methylation is not proper in any part of our DNA, that can activate the B cell and give the co-stimulatory signal. Okay, they can express and activate the B cell, T cell. Am I clear? So, DNA sometimes get endocytosed by this receptor mediated endocytosis, which interact with the TLR9 and can be activated to activate again by co-stimulatory signal. Normally it does not happen, but if it happens, then disease can also happen. You can see it is very unlikely that DNA is traveling around in the blood or in other tissue, because DNA is so inside the cell, it is so much protected. See, they are covered with histone and then nucleus, then nuclear membrane, then cell membrane. So, suddenly coming naked DNA into blood or exposed to the immune system is very rare. So, this is not a very common, but it can happen. So, now again what I was telling, the peripheral tolerance. So, this is the normal case. Okay. All the B cell receptor or T cell receptor which is interacting with the cell protein are eliminated, but we already discussed about hypermutation, and I discussed during diversity generation, and Professor Ganguly already discussed during B cell when it happened. You know what is hypermutation. So, B cell maturation or B cell immunity also you have heard this term. So, during hypermutation what happened, the CDR region change, right. So, we told that hypermutation make the B cell receptor more diverse as well as more specific, okay, better antibody, right. So, during that hypermutation, when change is happening, that change not only doing the good, opposite thing also can happen. By mutation they can become the auto antigen or self antigen specific. So, if it becomes self antigen specific after hypermutation, it was not before. After mutation it changed the specificity towards our own protein. If this happen, this single interaction or cross linking gives the signal to die. So, that cell will also die in lymph node or the secondary lymphoid organ. So, they also are not going to come or see the we will see them again. So, this is also a very good mechanism for and the self tolerance. Okay, but by any chance if this does not happen for any particular antigen, that B cell will be activated or T cell will be activated, and that will cause autoimmunity. Okay. Antigen in immunologically privileged site do not induce immune attack, but can serve as target. What does it mean? There are few places in our body like brain, eye, testes, uterus or fetus, they are immunologically privileged. It does not really mean that no immune system is there. So, T cell can go there, B cell can go there, but there are certain mechanism. Okay. So, brain has a blood brain barrier, not everything can cross. Okay. So, in uterus they have a placenta membrane or trophoblast. So, it is normally when baby is born our T cell, B cell cannot travel that easily. Okay. So, they have barrier, they have certain T regulatory cell which control the immune system. So, somehow these places are not very active site for immune reaction, that is why they are called immunologically privileged site. But what happened, by any chance suppose there are two eyes, one eye by some trauma or blow or some accident it is damaged. What happened? Then the antigen will release, and you know the dendritic cells or Langerhans cells are there. So, that broken cells or the antigen will brought to the nearest lymph node, where the reaction will happen, that we already know, I am not going to discuss. So, much antigen full of antigen of that originated from this retina or the eye or the ocular region come, and the T cell will be activated, and that T cell, activated T cell can go and damage both the eye. But this is not a normal case, but it can happen, and autoimmunity may be responsible for that. Autoreactive T cells that express particular cytokines may be non-pathogenic. Normally, autoreactive means all the subset of T cell, Th1, Th2, Th17. These cells are not doing any harm to us. Okay. They are non-pathogenic, but sometimes they also become pathogenic lymphocytes. They are suppressed because just in case if some similarity are there, they activated and then it will cause the problem. Autoimmune response can control at various stages of regulatory T cells. Okay. How? So, T cell specific for self antigen recognized in thymus. So, that is what we are calling. So, if the thymus that we just mentioned, if thymus it react with our own antigen, the natural regulatory become natural regulatory T cells. So, normally T cell regulatory cells, how it is developed? So, when T cells interact with or moderately interact with our own protein, it become T regulatory cells. Okay. This is in thymus, this is central regulation, and peripheral regulation also. If T cell specific or self protein or commensal microbiota that which is present in our alimentary canal and other parts of the body, which is helpful, that if it is interacting with that protein, that means it is not supposed to do, but by any chance if this T cell somehow released from not the regular T cell released from thymus to peripheral blood and interact with the same or similar protein here, there also the antigen presenting cell produce TGF beta, which make them T regulatory cells. So, either in the central system like thymus or in peripheral system, if it interacts with our own protein, then either by cytokines or there are also cytokines definitely involved. So, they become T regulatory cells, and I already told T regulatory means the negative impact is there. So, what happened? So, suppose this become T regulatory cells, okay, by interacting with our own protein. In this case, if some active T cells somehow they bind to this same same antigen or same antigen presenting cell interacting with this. So, what will happen, which is already T regulatory cell converted, it will produce TGF beta IL-10 and inactivate them. So, what I mean, so these T regulatory cells which generated in the central system or in the peripheral system can inhibit the T cells which is interacting with our own protein. This is a normal case. So, that means that is how we survive, that is always happening, that is why you do not see autoimmunity so often. Okay. So, these are the different thing like autoimmune tolerance and making and breaking. That means, these are the points by which self tolerance made. I am just summarizing what we told. If any of such thing are not function normally, that means some problem will happen, and that problem means autoimmunity. What are those? I am just going through quickly. A critical function of the immune system that we already discussed. Okay. So, that is our own system is what? Discriminate self and non-self. Multiple tolerance mechanism are there. What is that? This is peripheral tolerance and central tolerance, right. They maintain like T cell regulatory. Central deletion is the major point, like deletion of the auto and antigen reacting B cell and T cell, so that no B cell T cell supposed to interact strongly with our own. I am saying strongly. Initial part, when we were studying the diversity, T cell immunity, B cell immunity, their development, we were telling that deleted, but now we are saying which is acting strongly only, they are deleted, not all, because all, if all the B cell receptor will be deleted, self reacting, then our immune system will also be compromised. Okay. Lymphocyte that bind self antigen with relatively low affinity usually ignore them. So, normally they do not do any harm to our own protein, weak interaction or moderate interaction. Okay, only strong interaction can create problem, but do not do, but sometimes it is activated. The example I gave with the DNA. Antigens in immunologically privileged site, normally they do not induce the immune attack. What I gave an example with the eye, that the normally the immune system do not react much, but if there is any breakage or injury or the damage of the tissue, that can happen also. Autoreactive T cells that express particular cytokines, that is non-pathogenic, do not do non-pathogenic. Pathogenic means they do not do any harm to our own, but may suppress the pathogenic lymphocyte, okay, or may suppress the pathogenic lymphocytes. So, that is how we survive, but if there is any problem, again disease. Autoimmune response can be controlled by various stages and regulatory cells, like what I said, the T regulatory cell at the central, peripheral, or Th1, Th2, all there is a balance, okay, T17. So, that balance is very important, all are there, okay. They are supposed to fight for foreigner only, but sometimes if the foreigner and our own protein, foreign protein and our own protein are very similar, we cannot figure it out, right. I mean, you can see we are so similar. I mean, all suppose for example, there are some very rarely you can figure it out difference between all the Europeans, okay. But there are certain difference, very minute difference, but for us who are not habituated to see Europeans of different country for long time, we cannot figure it out who is from which country, okay. Even that is true for India also, we cannot suddenly say who is from which state, because if they look similar. So, that close similarity sometimes create problems, okay, and that can cause autoimmunity. Okay. So, for today this is all.